Oh boy, if you've ever wondered, hey, do I record these videos in one try no matter what? The answer is no. My last recording of this went way too long, and we're just going to jump in because time's a wasting. So, last one, prokaryotic regulation, this time eukaryotic regulation. Oh, what do I... Yeah, let's go. So, gene regulation in eukaryotic cells carries a bit of extra significance because we are well, not all are multicellular, but the ones that are have to have some sort of cell specialization or differentiation. All of our cells have all of the genes we need, even if we don't use them, but it's the differences in expression of these genes, so in what proteins we actually make, that leads to this cell differentiation, specialization, and we get these different cell types. The proteins that nerve cells make are very different than the proteins that intestinal cells make, and it leads to the cells themselves looking different and having different functions, but they do all have the same genome. Technically, they just don't all use the same parts of the genome. So when we're talking about chromosomes, and we're really talking about a bunch of chromatin, which is the DNA protein complex. It's just DNA wrapped around proteins. The proteins themselves are these things called histones. They're made up of 8 subunits, and they really just serve like beads to wrap the proteins around, wrap the DNA around and keep it organized and tightly together. It's like wrapping your headphone cord around your finger before you put it in your pocket so it stays kind of organized. Same idea here. We're just having something to wrap the DNA around so it's not all tangled and a jumbled mess. This kind of keeps it in one coherent string. But we can kind of dictate how tightly do we want to wrap that DNA around the histones. So we can chemically modify them, which we'll talk about in the next slide, but we can also just wind the DNA either tightly or loosely. So if we wind it very tight, this is heterochromatin, and because it's wound so tight, there's just not room for the transcription machinery to get in there and do anything. So those genes are just physically not accessible to all the proteins that we need to actually make and transcribe those genes. This is what the chromatin will look like pretty much across the board during mitosis or meiosis when it's in that X shape. It's very tightly wound. We're not making any genes off of it. It's kind of its most protected form. It's less likely to be mutated or anything like that, but we can't use it for anything either. Its counterpart is euchromatin, which is the active form. It is the form that chromatin takes when it is expressed and we're actually making the proteins off its genes. And again, it just comes to the idea that if it's loosely wrapped, there is room for the transcription machinery to come in and actually do its job without messing a bunch of other stuff up. So if it's tightly packed, we're not using it. If it's loose, we are using it and actually making the proteins. We can also chemically modify the histones themselves. So methylation is the addition of a CH3 group, basically just unreactive carbon. This serves to condense chromatin and suppress transcription because it can just pack in very tightly, and the CH3, the methyl, isn't really going to be reactive. Acetylation, on the other hand, is more reactive. Acetyl groups are COOH. They're acidic, they're polar, so they'll repel each other, and this all just serves to kind of loosen up the chromatin overall and therefore promote transcription. So in general, just think of methylating. Addition of methyl is always going to be silencing, and acetylation will be promoting. We can even methylate the DNA itself. This is something I'll talk about in class more, but we can methylate the DNA. This actually reduces transcription as well. It can do long-term inactivation, and the interesting part is we're finding it can be passed down to offspring, meaning what happens to your parents can actually affect you physically, which is crazy, but we'll talk about this in class. Main focus today is talking about the nitty-gritty of how we regulate eukaryotic genes, and it is all about space and just is stuff in the way, or how easy is it for RNA polymerase to get in there and do its job. So RNA polymerase needs help. It's kind of useless on its own. It needs helpers. It needs a bunch of other proteins to help it attach and actually proceed and do transcription well. So there are these things called enhancer sequences. These are sequences of DNA that are what's considered upstream of the gene. So it's not right next to the gene. It's not the DNA right before the gene. It can be pretty far away. It can even be on a different chromosome, technically. But these are DNA sequences that bind to activator proteins. They can also bind to repressor proteins. But they bind generally to activator proteins that then stimulate transcription by providing a bunch of binding sites for RNA polymerase and its associated proteins. Then there's the promoter sequences, and these are right next to the gene, very similar to the promoters that were in operons. They don't code for anything. They just serve as a binding site for transcription factors. So they can make it more accessible, less accessible. It depends. But your promoters are kind of the same as they were in operons. It's just the place where RNA polymerase is going to attach, along with a bunch of other transcription factors. So the main idea is that since these are all controlled by individual enhancers, they all have their own proteins. They all have all the enhancer proteins will have their own promoter sequences. And so there's a lot more ways to turn these on and off than there would be in a bacterial operon. Because instead of having just one main control switch, in this case, in these diagrams, I have 3 control switches each. Because, again, if genes are co-expressed, like the human lactase genes, are not all next to each other in the genome, they can be spread out, and they all have their own promoter sequences, so we can regulate them all individually. And then they can all have their regulatory effects on their genes. And the interesting thing is we don't have one circular chromosome like prokaryotes, so we often have these genes on completely separate chromosomes. Because if you think about it, 2 genes being on separate chromosomes doesn't necessarily mean that they are far apart physically in the nucleus. This image shows like if you gave each chromosome a color, where is it, of the 23, and it's less useful for me because colorblind again, but the idea is that if we have these genes that are going to be expressed together and they're all related, even if they're on different chromosomes, we can just put the loops with those genes and overlay them on top of each other, all in the same general region in the nucleus, and that makes it easier for all the transcription stuff to happen. So we can, even though they're on different chromosomes, we can overlay those chromosomes. So I get like chromosome 1, then 2, then 3 on top, and I can overlay them all in the same area in the nucleus so that they're still close by physically when we're actually using them. They're just not close by in terms of on the same chromosome or not. But again, activator proteins promote transcription, repressor proteins inhibit, and they both just attach to sequences that are not even necessarily next to the gene that we're looking at. It could be on a different chromosome. We also can do some post-transcription regulation. We've talked a little bit about this with RNA processing mainly, where you can make different mRNAs from the same DNA transcript. So this is showing the troponin T gene. There's 5 main exons that they number 1, 2, 3, 4, 5. And you can make 2 different mRNA molecules out of it, depending on which one you keep. If I keep 3, I get 1, 2, 3, 5, or I keep 4, I get 1, 2, 4, 5. I'll get slightly different proteins that can have slightly different functions. Again, lets me regulate even more. If I don't need the 4 version, I just don't make the 4 version. We can also stop the translation from even beginning. So regulatory proteins or RNA itself can bind to the mRNA that we made and just stop it from attaching to the ribosome and therefore undergoing translation. And even if it does undergo translation, the proteins themselves can get cut up, joined together, or even have chemical groups added like the iron in your hemoglobin for a functional purpose. So, for example, the hemoglobin, if you don't have iron, even if you make the hemoglobin proteins, you're not going to have functional hemoglobin until you have the iron. So it's another way to regulate with, again, do I add the chemical group or not, gives us more opportunities to turn the gene on or off. Last thing is these, or last new thing, are these non-coding RNAs. Most of our DNA doesn't code directly for a protein. Only a small fraction actually directly codes for a protein. And of that small fraction, a smaller fraction, even less than the genes that code for protein, are genes that code for special types of RNA. So the ribosomal, the transfer RNA. The types here are these non-coding RNAs. That's what ncRNA is. ncRNAs don't code for proteins. They instead regulate other genes, usually either with mRNA translation or how the chromatin is organized. But the main idea is like what these microRNAs do is they just bind to the mRNA that we made, mRNA transcript, and they either degrade it or they just stop it from attaching to the ribosome. And so they can regulate transcription that way. They're generally inhibitory, and they work just by blocking off the attachment sites and preventing the binding from happening. So to sum up, we have regulatory proteins and we have regulatory DNA or RNA, usually DNA sequences. So prokaryotes have no protein associated with the with the promoter sequence. No proteins go with promoters. The promoter sequence is just a spot for RNA polymerase to attach. They have operator sequences, which are the sites. They're kind of embedded in the gene, and they're places where things like repressor proteins can bind to and actually block RNA polymerase from continuing to transcribe. It's like putting a roadblock in the middle of the road. Then you have the activators, which bind to the activator binding sites. These are proteins that attach upstream of the gene and usually help RNA polymerase attach and keep the active site open for that. Eukaryotes are more complex. We have two main types of regulatory proteins. There's general and specific. So your general transcription factors are things that will just generally bind to the promoter sequences, usually containing a TATA box that just kind of work for all genes. It's just kind of the basic machinery you need to get RNA polymerase on and get everything going. Then you have specific transcription factors, which will be able to either enhance or repress or silence specific individual genes. So activators bind to enhancer sequences and serve to stimulate transcription. Repressors bind to silencer sequences. I didn't have them labeled as silencers. They were all labeled as enhancers. And they would serve to repress transcription. So if a repressor binds to a silencer, it blocks RNA polymerase. If an activator binds an enhancer, it helps RNA polymerase attach. And that is all for gene expression. Second try, I cut it down by 5 minutes. That's all you're going to get. I'm not doing a third try. We'll go over this in class. It is complex. I think gene expression is very, very, very tough. We're going to work on it in class. So please write down any questions you have, ask me, and we will get there together. That's all. See you later. Mutations is easier and more fun. This was the hardest. You made it. Congrats. Woo! No, don't start streaming. Oh, no.